So welcome everybody. Welcome everyone to tonight's presentation, updates on frontotemporal dementia with Dr. Finger. So a couple of housekeeping notes before we get started. So everyone's video and sound has been shut off, and we ask that you keep them off for the duration of the presentation. Dr. Finger is happy to answer questions throughout the presentation, as well as we will have some time set aside at the end of the presentation. So if you think of a question as she's talking, feel free to enter it at that point. And as I said, she'll take some questions throughout. So how to do that? There is a Q&A. So if you move your mouse or touch your screen, for me, it's at the bottom of my screen, but I know every device is a bit different. But there's one spot, it's got two little bubbles and it says Q&A. So if you click on that, you can type your question in there and hit enter, and we will see it and we'll get to those questions. We do ask that you don't enter the questions in the chat box. We kind of set that aside. If anyone's having technical difficulties or needs some assistance, you could enter that in the chat and we will monitor that. All right, so I'm pleased to introduce Dr. Elizabeth Finger. Dr. Finger is an associate professor in the Department of Clinical Neurological Sciences at the Schulich School of Medicine at the University of Western Ontario. She's a behavioral neurologist at Parkwood Institute of St. Joseph's Healthcare in London, Ontario. Dr. Finger's clinical practice and research focus on disorders of cognition and emotion, including patients with frontotemporal dementia and other neurodegenerative diseases and dementia that present with changes in behavior or language. So it is my pleasure to introduce Dr. Elizabeth Finger.
Great, thanks very much, Tara and Catherine, and hello everyone. Again, I think I said this last time, I'm really sorry. We're not in person, or at least able to be in a more naturally interactive format, but hopefully next year, and I think better than ever, and this format certainly offers some advantages for those that are at a distance or have other home care needs. Before I forget and get started, if we were in person, I was looking forward to introducing you to one of our new team members, Dr. Miguel Restrepo Martinez, who is in the audience tonight. Dr. Restrepo Martinez is a neuropsychiatrist and is working with our team. And so many of you who've come through the clinic or research programs in the past few months would have met him. And I'm thinking he may be a good person to bring back as a speaker in a few months' time. And there are a number of topics that he could speak about as well. So I'm just going to share my screen here. And I have to say, to me, it feels like time is just flying by these days. So it feels like not that long ago we were doing the last update. So with that in mind, I was thinking that we probably had a crowd that was pretty updated on the basics of frontotemporal dementia and dementia in general. But Tara and Catherine certainly reminded me we may have some newcomers. So I put just a few background slides in, but the rest of the talk is kind of different from last year. So I certainly invite you through the chat to ask questions if we've maybe jumped ahead of something and it's not clear to you where that fits in to the general topic or the issue at hand. And certainly at the end, we can welcome any question, even if it's a bit outside of FTD as well. So my obligatory disclosures, no commercial interests with respect to what I'll be talking about tonight. And so the overview. So I really did want to do this as an update. Both I and Miguel have recently returned from the International Society of Frontotemporal Dementia Conference that happens every other year in normal times. And so we've got some updates from the international FTD community there that we'll share. That includes some updates on clinical trials. And this time I've also really tried to give you an update on a lot of the research programs we're participating in locally or conducting locally, because I think many of you have participated, and so we'll give you a status update and let you know where research is continuing. So very quickly, we did this before, and I think the talk is online through the McCormick program if you didn't catch it last year and you just want a refresher on some of the basics. But this is the kind of technical definition of dementia that we use. The loss of cognitive functioning in typically more than one domain that is severe enough that someone cannot do the basic things that they typically are able to do. And so under this umbrella kind of functional description of dementia, we have multiple different causes that typically come on in mid to later life, with Alzheimer's being the most common, and that's why sometimes it's synonymously used with the word dementia, but really Alzheimer's is just one of many causes of dementia. Tonight, our focus is on frontotemporal dementia, as that's a focus of some of the caregiver and family support groups there at McCormick, and certainly is a large focus of our clinic and research program. But also under this umbrella of dementia are Lewy body dementia, the related Parkinson's disease dementia, stroke-induced dementia, which is called vascular cognitive impairment, and then there are a few other less common causes as well. These different causes of dementia, again, just as a refresher, are defined by the different pathologies or changes in the brain cells that we see if we look at someone's brain tissue after they've passed away. So with Alzheimer's disease, the findings of amyloid plaques, these round brown circles, and tau tangles, these darker, more linear or oval inclusions in the neurons, are the hallmark and are what defines Alzheimer's disease. In Parkinson's disease and Lewy body disease, we have a different molecule building up. It's called alpha-synuclein, and it aggregates in these round balls that were once named Lewy bodies, and hence the name of Lewy body dementia. And then for the frontotemporal dementias, pathology concentrated, like the name says, in the frontal or temporal lobes of the brain. And typically, most commonly, we see one of two abnormal molecular patterns, either aggregations of the molecule tau or abnormal aggregations of the molecule TDP43. Now, for all of these molecules across all three of these dementias, these are all molecules that we normally have in our brain and normally have in our brain cells. And so it's not clear why they start to aggregate or clump up in these ways, and whether the problem is with the processing of the cells and the clumps are toxic, or whether these aggregates are not toxic themselves, but merely reflect a process that is toxic upstream or downstream to the neurons. And that's still a big area of research. Very basic demographics, frontotemporal dementia is roughly evenly split in terms of the number of patients affected who are male or female. And there are three main subtypes of FTD, the behavioral variant being the most common, and then two language subtypes, the non-fluent progressive aphasia or the semantic variant progressive aphasia. This is one example of what a brain scan that is supportive of a diagnosis of FTD may look like. Here we are looking at a scan where we are taking or seeing slices in this plane. So it's as though the patient's feet are coming out towards us. The white you see on the scan is the normal fluid around the brain. And if we draw that line between the front half of the brain and the back half of the brain, you can see that the spaces in the front half are much larger than any of the white spaces that we see in the back of the brain. And so this is reflecting atrophy or shrinkage of the frontal lobes, the insula, and part of the temporal lobes, which is hallmark of frontotemporal dementia. Just another scan showing the temporal variant. So sometimes patients have a lot of temporal atrophy and less frontal atrophy. This is the temporal lobe here. And you can see this little string of tissue that I'm outlining is almost like a string, as I mentioned, as opposed to the nice round normal appearing brain more posteriorly. So you can really see how focal this illness is and how it really are only certain regions of the brain that are particularly vulnerable to the damage this disease causes. This is a picture of a brain after someone has passed away. The pathologists remove the brain, and here you can see that the front half of the brain, even just on gross visual inspection, shows much thinner atrophied cortex compared to the right 2/3 of the picture, which are more posterior parts of the brain, which are normal in size. So again, frontotemporal dementia named for the region of the brain that it affects, and it really often does so with a striking focality. So causes of FTD. So this is a slide from a NICE review just from 2 years ago, reminding us that about 25 to 45% of FTD appears to have some hereditary component, while the majority of patients with FTD appear to have a sporadic version. So 50 to 60 or even 70% have no family history of something that is suspicious of FTD. For the familial forms, we can see 3 main genes that are associated with causing FTD. So a mutation in the C9orf72, the granulin, or the MAPT gene all can cause frontotemporal dementia. For the sporadic patients, most of the time, if we do genetic testing, we don't find a gene. At least 80% of the time, we don't. However, even when the family history appears pretty benign, we can see that these FTD causing mutations are found in a small percentage of patients. Most commonly, the C9orf72 might be found in up to 6% of patients with sporadic appearing FTD, with lower percentages for other FTD causing genes. And so this is why when you come to the clinic, we talk about whether the family history suggests a familial or sporadic form. And then if we do think that it is hopefully likely sporadic, we still talk about the possibility of having clinical genetic testing, which is available in Ontario through the Ministry of Health. This picture just shows you that FTD is an international or worldwide disease, and this is showing the prevalence of the different types of mutations in FTD research cohorts to date. So at the very bottom of the slide, you can see that they've color-coded those carrying the C9orf72 mutation in yellow, the MAPT mutation in blue, and the granulin mutation in orange. And so, if you are thinking about this in your family, or it is in your family, you can think about where your more distant relatives prior to living in Canada may have lived, and what the patterns are there. And I'll say what is new and what is an update to share with you is that while much of the original research on FTD was really concentrated in Europe and in Canada, there's really been an explosion of FTD-related research and clinical programming worldwide. So that includes exciting new programs and research activities in South America, in India, where they've been found to have a higher proportion of MAPT carriers, and China and Japan, and Australia and New Zealand now with their own FTD cohorts and genetic FTD cohorts as well, which will really accelerate, I think, understanding of these conditions. This was another update slide. So a lot of times in the clinic, especially if we're talking about a pattern that looks like familial FTD, or we know that it is a form of genetic FTD in the family, we will try and think about, well, at what age is someone likely to develop symptoms if they carry the mutation? So this was a study that was published from data with the GENFI study, Genetic FTD study, which we participate in, and which has sites across Canada and Europe. And they combined data with a really large genetic FTD cohort from the US as well. And so these graphs show for each of the gene mutations how the age of onset in the individual patient correlated with the age of onset in their parent, which is the column of graphs to the left, or how it correlated with the mean age of onset in their family. So not just the parental age of onset, but if there were aunts, uncles, grandparents, cousins that were affected. And so for those that aren't used to reading graphs all the time, what we're really looking at is the slope of the line that is drawn as the average relationship between these two parameters. If the line were completely horizontal and flat, that would tell us there's no relationship. And the more slanted it is to be on the exact diagonal, the stronger the relationship. So the take home from this is that while there is some correlation with age of onset, for each of the 3 gene mutations for a patient and the mean age of their parent or their family member, the strength of the association is somewhat weak, especially for the C9orf72 mutation and the granulin mutation. So the summary is, it is hard to predict what age someone who carries a mutation may show symptoms, even if we know the age of onset in their other family members or their parents. On the genetic side, of course, we've known about the 3 most common genes now for a while, but the list of genes that now are known to be associated with frontotemporal dementias is ever growing. At the meeting that was just a few weeks ago, we learned about several new variants, one of which really caught our attention, the CYLD. This is probably going to be very rare, but it's interesting because mutations in this gene have been seen in patients who have both tau and TDP43 pattern pathologies in the same family. And that's really intriguing because, again, the field still doesn't know, are those different aggregates toxic, or is it some upstream process that's toxic, and those are just sort of the downstream signs of it. So we are kind of always interested in if there are shared missing links connecting the tau and TDP43 pathology, sorry. We have had a couple of families in our clinic in London who have had some family members with tau in some and TDP43 in another. That's pretty rare. So we are looking forward to going back and rescreening those samples for this new CYLD gene to see if that might explain some of the inheritance in some families. Other updates from the meeting this year on the genetic front. So, when we talk about heritability in FTD, we're mostly talking about the behavioral variant or the non-fluent aphasia. The semantic variant of FTD tends to be less hereditary. It's usually only one person in a family across multiple generations that gets semantic dementia. And so the geneticists, when they think about whether they have anything to add to our understanding, have started to wonder if there might be what are called somatic mutations in semantic dementia. So those would be mutations that are not present in the egg that comes from the mother or not present from the sperm that comes from the father, but are mutations that happen in a subset of the cells in the embryo and later the individual that forms. And so some of their cells may carry a mutation and some may not. And that would be one reason why you might not find mutations in their blood. They may only have some mutations in certain cells. And in this case, the hypothesis is maybe in patients with semantic dementia, there were some somatic mutations that affected cells in the temporal lobe of the brain that is most affected by semantic dementia. So they don't have results yet, but we're really eager, to hear what comes of that interesting idea, and we'll bring those updates to you in the future. The list of potentially protective genes as well as risk genes is growing as well. These are genes that on their own don't appear to cause FTD or necessarily be 100% protective, but they do seem to influence slightly the likelihood of developing disease or influence the age of onset of symptoms for someone who develops FTD and has another causative gene linked to their FTD. And then finally, the other sort of update we were hearing in the meeting today is the look for recessive mutations. So, there are 2 main classes of genetic mutations that cause human disease. One group are called dominant, and that means you only need one version of the mutated gene on one of your chromosomes to likely develop the disease. And so those are the ones that tend to show up in every generation because you only need that one copy from your mom or dad to get it. Recessive mutations are different. Recessive mutations are mutations in genes that result in a disease only if you have 2 copies of that version of the gene or 2 copies of that mutation. So you would have had to get that mutated gene both from your father and from your mother. That means that recessive diseases tend to be much rarer, and they're also harder to identify because you may not see either that family history or that pattern of disease in the parents or even in the grandparents. It may look like a sporadic disease. So no recessive mutations found yet to explain what looks like sporadic FTD, but again, this will be an area we will follow and bring you updates in the future as they work that out. On the clinical side, the picture on the left is just a screenshot of the website. This is the dementia genetic panel that we are able to access in Ontario through the clinical pathways from the Ministry of Health. And you can see there are about 18 different genes on here where mutations can present like a frontotemporal dementia or present as frontotemporal dementia, and a couple of these for Alzheimer's disease as well. So we do have a very nice program here in Ontario funded by the government that helps us to get most of the well-validated genes relevant to these conditions. So while in genetic forms of FTD, we can be quite confident about the diagnosis and the cause if we find a gene mutation. And in sporadic forms, if there is that hallmark shrinkage or atrophy in the frontal lobes and the right behaviors or language changes, we can be confident about a diagnosis of sporadic FTD. But we still really benefit both clinically and from a research perspective in the ability to look at a patient's brain tissue after they've died to know more of the details and understand better what their most specific diagnosis was, and to learn about new patterns of changes in the brain that give us clues to the causes of these diseases and may point towards treatment. So this is a repeat slide from last year as well, but it remains, excuse me, very true today. What you see at the top with that rectangle labeled BVFTD is the rainbow reflecting the different patterns of pathology. So that's the different changes within the brain cells that you can see in patients who have a clinical presentation of FTD. So a lot of different molecular patterns, as you see from the rainbow there. And the chart below just breaks them down into the tau patterns. So not only is there a tau FTD, but there are different subtypes of tau abnormalities that are associated with different behavioral and symptom presentations of FTD, including cortical basal syndrome or cortical basal degeneration and PSP, progressive supranuclear palsy. Pick's disease may sound familiar here. We used to call all of FTD Pick's disease. Now we call it FTD, and we save the Pick's disease for this specific pattern of tau in the brain. TDP43 also has multiple subtypes of patterns, and more recently discovered the FUS or FET pattern of molecular changes, where a lot is still being worked out. And some of the updates we heard about a few weeks ago, we're trying to understand the clinical features of this group and potentially looking for gene mutations to explain it. Patients with FUS-associated FTD tend to be younger at age of onset. They tend to progress quite rapidly. And so far, there seems to be a bit of a male predominance. So with that, I wanted to give you a bit of an update as well on our local program and research that helps to support research using brain tissue in patients, both with frontotemporal dementias, but also Alzheimer's disease, Lewy body disease, and concussion. So this was had a soft start a couple of years ago and a formal launch about 1 year ago as the Daily Creighton Brain and Biobank of London, Canada. The website is here, and certainly we'd be thrilled for those that are interested. If you check out our website, there's a lot of information there. Overall, we consider this a nice curation or a biorepository that is leveraging all of the hard work done by our multiple clinical teams, by our patients and their family members as they've had their symptoms and diagnoses evolve, and all of the testing, including radiology, blood, CSF when it has been collected previously, and other assessments that they have done. So I'll show you in a slide, where we are with that, but certainly we always welcome patients or family members on behalf of patients if they've been followed through the London clinics or hospitals who would like to donate their brain after death, who have one of the diagnoses here on the screen, and we can certainly give anyone interested more information about that later. We curate this data and these samples, and we then make them available to researchers, both locally, but also throughout Canada and internationally, to try and accelerate research and discovery in these conditions. So we've had a recent tally of our patients who have very altruistically donated their own brain tissue samples and data to the brain bank, and some of these were brought in because they had been previously done, and others have been enrolled in the past few years with a formal launch. So we already have 145 patients with the frontotemporal degenerations, and you can see some of the subtypes here. Quite a few patients with Alzheimer's disease and Lewy body disease, and then various combinations as well. So we increasingly recognize that it's not always one disease explaining a dementia. Sometimes a patient has both Alzheimer's and Lewy body disease at the same time, or vascular cognitive impairment and Alzheimer's disease at the same time. And so there's a lot of research to be done on how those diseases might interact and influence each other as well. I'll just pause here maybe before we go on to the next topic. Catherine, I think you're monitoring the chat, but certainly welcome if there are any questions along the way for people to submit them, as we'll cover a few different topics and it might be hard to remember what your thought was at the time.
No questions so far, but I'll let you know if any appear.
Okay, great, sounds good. So we want to shift gears a fair bit for a moment, as you'll see in the title here, to talk about an idea that's been gradually maybe kind of simmering in the field, and that we in London are now poised to lead an international cohort looking at this. So we're talking about our neurodegenerative disorders, only disorders that start in midlife or later life? Or is it possible that some of the things that happen as our brain develops set us up for these diseases later on? And one of the easiest ways to try and understand this and to learn whether there may be neurodevelopmental origins to neurodegenerative diseases is to look at patients with genetic FTD and look at cohorts of individuals at risk for genetic FTD. So this idea, as I mentioned, has been simmering in the field for a while, and it probably maybe first was sort of put out there formally by Marcel Mesalom and Sandra Weintraub at Northwestern University, who are leading clinicians and investigators in the field of FTD and the primary progressive aphasias. And they happened to notice that many of their patients who developed a primary progressive aphasia, this disorder of language in midlife or later life, actually had learning disabilities related to language in childhood. So, normal IQ, normal functioning, but perhaps had some dyslexia or other language issue. And so it got them wondering, were those things happening in early life and later life connected? The Northwestern group followed that study up later on and indeed found that patients with primary progressive aphasia had higher frequencies of learning disabilities, and so did their first degree relatives as well, indicating that there was probably some hereditary component to this, and that perhaps, at least in some patients, it made those language areas vulnerable later in life. More recently, now that more of the genes, mutations that cause FTD have been discovered, are known, and we've been able in the field to study patients and families. There have been a number of studies now showing that kind of going as early as patients' 40s or even their 30s, that there are some subtle differences sometimes in those that carry a mutation versus those that don't when they first enroll into these studies. So, whether their first visit is at age 30 or 40 or 45, in some studies, we're seeing there are already some structural brain differences and sometimes possibly some very subtle cognitive differences, though often not. And then one other intriguing kind of clue that made us think about looking at neurodevelopment was that in families who are known to carry the C9orf72 mutation, it's kind of been observed clinically, and now it's been validated in a study that their family members seem to have slightly higher rates of what are considered neurodevelopmental brain conditions. So that might include schizophrenia or autistic spectrum disorders. And so it also raises the question, well, are those individuals carrying C9 mutations, and does having that C9 gene version influence how the brain develops? And maybe that's important to know so that we understand better why those circuits start to undergo neurodegeneration later in life. To look at that, we have been analyzing some of the data from the genetic FTD study, the GenFI study. We're a site as well, and if any of our participants or families are here, thank you very much for your participation. These individuals come once a year, they tell us how they're doing, we talk about potential symptoms, they do cognitive tests, MRIs, blood work, and an optional lumbar puncture. We decided to look at the youngest people in the GenFI cohort. So those are adults ages 19 to 29 who have a known mutation in their family. Typically it's in their parent or grandparents. And typically these young adults don't know whether they carry the mutation or not. They're at risk, but they may not. And similarly, both myself and our research team are unaware of their genetic status. We're blinded so that there's no bias, and they don't have to know their status unless they're sure they want to. When we look at the GenFI participants worldwide in this young adult age range, we've got a mean age of 25, which means that they're about 50 years or more from the typical age of onset of symptoms in their family. When we ask these young adults and we ask their study partner, which is usually a spouse or a sibling, about different symptoms that we can see in FTD, we do not see any differences between those that carry the mutations and their family members that don't. So the rates of symptom reporting of behavioral symptoms, what are called neuropsychiatric, language, cognitive, and motor, are not statistically different. However, when we actually look at their brain structure and their MRI scans, we are finding some hints of some differences that might be intriguing clues about some early effects. When we look at those young adults who carry the C9 mutation compared to their family members that don't, we find that their total brain volumes are already a little bit smaller, those that have the mutation than those that don't. And we find that a structure in their brain called the thalamus is also a little bit smaller. The thalamus is known to be one of the areas that shrinks earlier on in the C9 associated FTD. And this study is telling us that very subtly that might already be the case. Their thalamus might be smaller and their total brain might be a little bit smaller, even by their mid-20s. And so what we don't know is, well, did it develop small and maybe it's kind of stayed that size, or did it develop of a normal size and then start to atrophy or shrink? And what that pattern is, has important implications for what we understand this mutation is doing and when we might need to intervene. We didn't see any differences in how they performed on the cognitive tasks, though. So although there might be these subtle differences in brain structure, those that have the C9 mutation are performing just as well as those that don't. In our MAPT mutation carriers, we see a little bit of a different pattern. So interestingly, those that carried the MAPT gene have larger total brain intracranial volume. So what that refers to is the size right below their skull. So it's not necessarily the brain volume, but that intracranial volume reflects how big their skull developed during their youth. Usually that kind of finishes developing in early adolescence. And it's an indicator for what was probably their maximum brain size, because the skull growth is really driven by the brain growing underneath it. So a little hint that maybe the brains of our mutation carriers were a little bit larger than the non-carriers at the peak of development. We found, interestingly, that those that carried the MAPT gene actually had higher levels of education than those that didn't. Also, perhaps indicative of improved brain capacity. And when we look at some of the cognitive tests they did in the GenFi study, we found that they performed better than the non-carriers on two tasks: a task of attention, which is the number of digits they can remember in a row, and on a task that asks them to generate as many words as they can think of, starting with different letters. They, on all the other tasks, performed similarly to the non-carriers, so they weren't worse on any tasks. So really interesting hints here that perhaps having that MAPT mutation in early life might even be advantageous. It may be associated with larger brain size, and it may be associated with higher brain function. Really intriguing, and may again help us to understand whether there are changes in these networks early in life that are advantageous, but maybe later on are a setup for vulnerability. We found a similar pattern in the granulin gene mutation carriers who are young adults. Just like the MAPT carriers, those that have the mutation have a larger intracranial volume, so larger volume under the skull. And they also had a larger volume of one region of the frontal cortex, which is the cingulate cortex. So again, maybe having the mutation confers some tendencies towards larger brain. And they performed better on one task. This is kind of a speed of processing task. The mutation carriers of granulin perform better than their familial non-carriers. So really intriguing to us and have prompted us to be quite interested in understanding, is the presence of these mutations having any effect early in brain development? And is that effect positive? Maybe is it advantageous, or is it already disadvantageous? So with that background, we have gotten funding from CIHR and are just now launching the GenFi neurodevelopmental study, or NeuroDev for short. And the idea is just that, to determine whether these genetic mutations that cause familial frontotemporal dementia might have effects on brain structure or function that we can detect in youth. The study will be similar to the GenFi study, that youth from our GenFi families who want to enroll and whose parents want them to be enrolled can come into the study. Neither the parents nor the youth would find out their mutation status, and the youth absolutely would not, and neither would I, neither would the study team. So everyone is completely blinded for very obvious and important privacy reasons there. The youth will complete similarly some cognitive testing, an MRI, a saliva sample, if they're willing, a blood draw, and we'll see them after 2 years and determine whether or not we're seeing any of these differences in those that may carry the gene versus those that don't at these younger ages. Certainly, at this meeting and most of the other FTD meetings, we haven't talked about kids before, and we recognize that's a big shift. in our thinking and practically, and we've given a lot of thought to working with youth who might be at later risk for FTD, though maybe not for 50 or 60 years. So we've done a lot of consultations with FTD patients, family members, participant engagement groups, the Association for FTD that many of you will know. We have made sure that our core team, in addition to our usual FTD team, includes clinical pediatric expertise, both from genetic counseling, neuropsychology, and developmental neuroscience. We're following a really nice model that the field of Huntington's disease has used when having a similar youth study. So they're quite ahead of us, and it has been well established now in Huntington's disease that there are changes that happen to the brain in neurodevelopmental periods because of the presence of the Huntington mutation. So in their youth cohort studies in Huntington's disease, they have all discussion of the disease and the risk be done by the parent with the child. So only what the parent thinks the child should hear, is ready to hear, or that the family, wants them to hear, completely controlled by the parent. So that is the approach in the GenFi NeuroDev study as well. Any questions the child may ask about FTD or dementia are referred back to the parent for the parent to handle as they would like to. The only thing the child needs to know for sure to be eligible to come into the study is that there is a history of dementia in the family. But details beyond that, again, are left to the parent's discretion. And then finally, we have many steps to ensure that, in no way could the child's identity and any of the data collected in the study ever be associated with that child. So multiple steps to safeguard that. And we don't expect to release data for many years until we have enough children that when it's released, there's no way anyone could figure out who they were based on their age at the time of participation or what language they spoke or anything like that. So we have 9 sites and probably adding some more sites as well after our presentation a couple of weeks ago. So it will be an international study, and we'll keep you updated in the years to come on this, of course. We've tried to make it fun for the kids as well. We've used tasks that are used in other youth cohort studies, so we know the kids can do them. And a lot of them are on iPads. They go pretty quickly, and our piloting so far tells us the kids find them pretty engaging. And I touched on it in my remarks earlier, but just, to emphasize the reasons that we think this is important. So we may identify that there are early compensatory processes, so early beneficial processes that are happening in the cells and in the brains of young people who have this mutation that protect them against showing any symptoms of FTD at that stage in life. And if we can understand what those processes are, those may be ways, therapeutic targets to support and boost those same compensatory processes through middle age and through later life. It will, perhaps have other effects if we were to find that there were differences. It may influence the clinical trial design and at what age we think adults should start treatment if they know they carry a mutation. And certainly if we find that there are any consequences that aren't beneficial for kids, that may also help to identify other interventions that can help them reach their full potential and maintain it. So maybe I'll just do a little pause there as well before we go into the next section, in case there are any questions right off the bat on either of the prior two.
So far, the only question was whether or not the slides and or the recording will be available. And yes, we are recording this session and it'll be available on our website soon.
Great, thank you. Okay, so with that, another area of interest of ours locally and certainly in FTD generally are the symptoms of FTD that I know, probably almost everyone in this group on the call or on the Zoom has encountered up close and frequently. We've been particularly interested in the symptoms of apathy and empathy deficits in FTD, in part because they're very common. They're quite disruptive to the normal relationships. Again, I don't have to tell you that. I know you know that. And we focused on them because there really aren't any treatments across any conditions for apathy or empathy that are effective yet. I like to give some examples when we talk about apathy of what we mean. And when it presents in FTD, it can be a loss of motivation, a loss of physical activity, a loss of interest in jobs or hobbies, a loss in interest in family or friends. And it certainly can overlap a bit with empathy deficits. So these are some of the more extreme examples that we have observed in patients in our clinic. But as you can see at the bottom, not only is it the patient with FTD doesn't realize their spouse is having a heart attack, and they just ask, well, what's for dinner? But it's every day. There are just continual manifestations of them no longer showing an interest in other people around them, turning very inward, becoming very self-centered. We often use the analogy of kind of like a toddler, that really their needs in that moment are all that are on the forefront of their brain, and sometimes all they really seem to be able to understand or represent at that time. So to try to find treatments for apathy and empathy deficits, we had done a series of studies that we presented last time, and I hope the recording is still available to you. But we got quite interested in a hormone called oxytocin. It's a nasal spray. It's a hormone that's in the brains of men and women. And in some animal models and in autistic spectrum disorder, it was getting some attention because there were some studies suggesting that it was boosting pro-social behavior, so boosting different kinds of interactions between people and between other animals as well. We had done a couple of pilot studies, and we were seeing an interesting signal saying perhaps it was improving apathy and kind of social interactions in some patients who were trying the nasal spray. So we then were able to scale up to a formal randomized clinical trial, a phase 2 trial, aiming to enroll about 100 patients and have them get a 6-week course of the oxytocin sprays. We compare the same patients also get a 6-week course of the placebo sprays, and we can compare how their behavior and how the apathy and empathy were during the treatment phase compared to the placebo phase. So we've called this study FOXY for short, for Frontotemporal Dementia Oxytocin Study. It was a bit delayed because of COVID. The enrollment really slowed down for a while, but picked back up enough that we are nearly complete. So our deadline for enrolling the last patient anywhere in the network of sites across North America is coming up this January 31st, 2023. That'll mean our last patient enrolled will be in June. So we expect to have results of that study maybe in 1 year or a little short of that. So for next year's update, I look forward to sharing with you what we've learned from that study and whether it's been effective or not. And just, many thanks to anyone who may be in the audience today who helped their family member or partner participate in the FOXY study. We think whether it works or not, it's important to figure that out. And we expect to learn a lot about FTD and help future clinical trial design through the FOXY study as well. If you do have a patient, family member who you think might be interested in this study, we do have space for just a couple of more enrollments locally. So I'll give you Sarah's contact at the end in case you want to reach out to her. She's our research coordinator.
Just wondering, Dr. Finger, if this would be a good point to pause there. Just 3 questions. The first one is, how widely is genetic testing available in Ontario/Canada?
Yeah, so I can speak to Ontario. I can't speak to Canada as broadly, but in Ontario, the ministry does cover genetic testing for a symptomatic patient who has a clinical diagnosis of FTD because of that, up to 40% potentially having a hereditary component. So you would just, need the patient to see someone in clinic who is familiar with that and knows how to order the appropriate testing and counsel about it. The other pathway is to get the patient referred to a genetics counselor. There is not necessarily coverage of genetic testing for other dementias like Lewy body disease, or not for most patients with Alzheimer's disease, because those are much less strongly hereditary. There is very rarely a single gene mutation that causes Alzheimer's or Lewy body disease. So there you would need a much stronger family history coming on early in life, typically in the 40s or 50s in multiple family members.
Next,
Mhm.
sorry, did you have more for that question?
Nope, that was it. Thanks.
Just two more. Second question, does diet influence the progression of dementia?
Yeah, it's a big question. So we don't know for sure. And the reason is there's really never, there hasn't been, and probably there couldn't be a study where individuals, from the same families are randomized to different diets, basically from childhood. So we always have confounds of, well, what was their diet and what were the other factors in their lifestyle? Our evidence for diet comes from these then retrospective studies. So they look at cohorts of people and try and identify, well, what factors or what diets might be associated with lower rates of dementia or later age of onset. From those sorts of studies, kind of the current best evidence suggests that the Mediterranean diet, and I'll explain that in a second, but that the Mediterranean diet may be associated with a slightly lower risk of developing dementia. Now, that's probably related to a lower risk of developing strokes and therefore a lower risk of developing vascular dementia, maybe a little bit lower risk of developing Alzheimer's dementia. We don't have any dietary evidence, not that I would consider strong, for frontotemporal dementia or Lewy body disease. So there it would be even less clear. But the Mediterranean diet, it's high in fruits and vegetables, fishes and legumes, lower in red meat and saturated fat. And so it's a diet that has good evidence for being better for cardiovascular health. So it's a good idea for anyone to adopt just for general health. It's not calorie restrictive, but it's just the kinds of foods that one should focus on.
Two more, and one more question landed. The next question, for the oxytocin clinical trials, what are the criteria for participation? This participant in our webinar states, I assume early onset, question mark, looks like stage 1 and 2.
Oh, sure. Yep. And sorry, some of that on the slide is a little bit of shorthand, but we had 2 stages of the trial. The first stage was where we were enrolling 60 patients to find the best dose. And now we're in the second half of the trial, stage 2, where we think we know what the most promising dose was. And we just need to enroll about 20 more patients, now even less than that, to try and determine whether it's effective. In terms of the eligibility criteria, they are designed to allow patients with FTD, whether it's behavioral variant, semantic variant, or the non-fluent aphasia variant. So any of those 3 who are in mild or moderate stages of disease. So they need to have a mini mental status exam score of 10 or more, and that's a pretty low score. So most patients with FTD who are living at home and still communicating and able to understand a bit, most of them will score higher than that. And then there's a general severity score that would be sort of moderate impairment. So, able to do most of their basic things like feeding themselves, using the bathroom, but certainly could be impaired where they need help organizing, banking activities, day-to-day things. And then they also need to have the presence of apathy. So apathy that we grade as kind of moderate. And there's a phone questionnaire that Sarah can do quickly, if someone is just wondering whether or not their apathy may be eligible.
And one last question for now. Are hallucinations associated with frontotemporal dementia?
Yeah, so interesting question. Yes, with some forms. So in particular, we now know with the C9 mutation that hallucinations, in particular visual hallucinations, and what are called tactile hallucinations. So the abnormal sensation that you have bugs crawling on your skin all the time, or you have water running underneath your spine, those kinds of sensations are common in the C9 group. For their visual hallucinations, it's often faces or people, but could be bugs or other visual kind of distortions. We used to, in the field of dementia, when we heard about visual hallucinations, we used to really think only of Lewy body disease or Parkinson's disease, because it's been known for a while there that patients may have visual hallucinations of people, children, babies, animals, and certainly some other kinds of phenomenon. Now, when we hear about visual hallucinations, we would think, more likely Lewy body Parkinson's, or could this be an FTD, and thinking about the C9 or F72. You can have some hallucinations in other forms of FTD for sure. They're just not quite as common.
That's all the questions for now. Thank you.
Okay, great. And I think I'm almost to the end of the slides here. One more. So one more topic related, but just another approach that we're taking. So, we mentioned that apathy is really common in FTD, and we don't have treatments for it. So it's different than depression. It's not that sad mood, but it's really kind of emotional flattening, a loss of the experience of pleasure for activities that used to give rise to that positive feeling or sensation. It's a lack of motivation to engage in kind of goal-directed activity. So patients who are apathetic may be content just sitting in a chair for hours at a time. We've had people stare at a wall or stare at the TV, but they can't even be bothered to turn the TV on, even though they know how to. So we've been thinking, in addition to the oxytocin, about why can't the field identify better treatments for apathy? And we think there are probably a few different reasons. One is that we put all kinds of different behaviors we observe under this umbrella term of apathy. And it might be that we need to really look at different types of apathy because maybe they need different treatments. So as an example, with the middle picture, we think about, well, might some patients have apathy because the parts of their brain that are normally sensitive to reward and anticipate reward or process reward are damaged. So things that used to be rewarding to them no longer are, and therefore they're no longer motivating. Or perhaps some patients have apathy, maybe their reward system is intact, but maybe the part of the brain that judges the effort that is required for a task is affected by the disease. And so maybe their brain is miscalculating effort as being much higher than it really needs to be. And that's why they don't get up off the couch to get a snack, because their brain has just said that is way too, energy costly of a task, or way too hard of a task. We're not going to do it. And then a third idea is that, well, maybe their reward processing is okay. Maybe their effort calculations are okay, but maybe they're just having trouble generating options of things to do. Maybe the part of the brain that does that is failing. On top of that, it might be tricky to find one treatment for everybody because we have different versions of genes that relate to some of the medications that are available, and we don't usually model what those versions are. So maybe some people with a certain gene would respond to an intervention and others wouldn't, and maybe we need to account for that in our clinical trials and our treatment models. and then the patterns of brain atrophy. So I showed you at the beginning one patient who had a lot of frontal lobe atrophy, also a different patient with a lot of temporal lobe atrophy. In clinical trials for apathy to date, all of those patients have been in the same group. But maybe, the temporal atrophy patients maybe would respond to one medication, but we're not seeing that in the larger group because the effect is diluted by inclusion of patients with trouble in other brain areas, and the effect is washed out. So our group, led by one of our graduate students, Ruby Malik, is trying to do that kind of parsing of apathy to see, okay, in patients with different causes of dementia, both FTD, Lewy body disease, Alzheimer's disease, vascular dementia, are we seeing different patterns of cognitive impairment that seem to be associated with apathy? So we're just at the beginning of looking at some of this data. And again, many of you may have family members or friends who participated. So thank you. This is the Hyades study. And so what Ruby has found in the first data she's looked at, that she has a task called option generation, where she presents the patient with a scenario like, you've just woken up in the morning, you can see the sun shining through the window, what are you going to do during the day? And patients have to freely generate different options that they could think of. Other scenarios are a little more specific, and so maybe only one or two actions would be appropriate. And so she looks to see how many options patients can generate. And if we group patients by those with dementia who are showing apathy in their everyday life, compared to those that are having dementia but not very apathetic, we find that those that are apathetic cannot generate nearly as many options to these scenarios as those that don't show apathy. So early data suggesting that apathy is associated, at least in some patients, with trouble in generating ideas of what to do. So even though they might be capable of, doing a drawing or washing a dish, the idea may not be occurring to them. In the graph here, she's looked at then this by the different diagnosis. So how many options of things to do could patients with Alzheimer's disease generate compared to those with frontotemporal dementia, with Lewy body dementia, and healthy controls? And so you can see in comparison to healthy controls, patients with Lewy body disease cannot generate as many options of things to do. Our patients with FTD are worse, and kind of interestingly, our patients with Alzheimer's disease had the most trouble in generating options. So this does point to some treatments, some of which we know work, like behavioral interventions of not thinking that the patient will come up with an idea and initiate it, but really being more directive and saying, here's the jigsaw puzzle, why don't you sit down and do it? Or here's the vacuum cleaner, why don't you vacuum this room, to overcome this trouble with option generation. Ruby has designed a couple of other really neat tasks to look at the other aspects. So the task at the top is looking at how much cognitive effort are patients willing to expend. So she has a visual search. You have to figure out which animal on the screen doesn't have an identical pair. And then there are different kinds of reward that the patients, are told they'll receive with each trial. And so they decide whether they're going to do the task or not, which is one measure of effort. And then whether they spend time on it and actually get it correct, which is another measure of effort. So with this task, we'll kind of see, are there differences in effort sensitivity that are correlated with apathy? And we're also looking at reward sensitivity because in some trials we have money, others we have candy or some kind of positive social videos. So we can also look at, are the reward systems affected? And if they are, is it all kinds of common reward, or is it specifically certain types of reward? And that might be useful behaviorally or in developing pharmacologic treatments as well. And then the task on the bottom is very similar, but instead of cognitive effort, this is actually indexing their physical effort. So it's a hand grip, a dynamometer that you have to squeeze really, really hard to get the red bar up to the goal line. So we can look at both cognitive effort and physical effort and see how those might correlate with patients' apathy and whether we see subgroups that might need different treatment approaches. So I've touched on our clinical trial for oxytocin in FTD. I just wanted to mention a few other trials that are going on now, because I think for a long time in FTD, there were no clinical trials because the understanding and knowledge in the field wasn't there. We weren't ready for it. But in the past few years, we've seen a growing number of trials targeting symptoms of FTD. And now, really excitingly, treatments that are trying to slow or ideally stop the disease or prevent it. And so we do think this is kind of a really welcome pivot in the field and a nice opportunity for our patients that wasn't there before. We are expecting in Canada a new clinical trial of nabilone, which is a synthetic cannabinoid, so related to cannabis, but a synthetic form. That's going to be a clinical trial for patients with FTD who have agitation to see if this may help compared to our usual standard of care. There is also ongoing and scaling up a speech therapy clinical trial for patients who have one of the primary progressive aphasias. This is led out of Northwestern right now, but it's remote speech therapy. So even now we can refer patients here in Ontario to that trial if they're interested in doing the online remote therapy. And they were just funded for scaling up that trial. So that will be continuing in an even more extended way in the near future. And not locally, but certainly internationally, we heard recently about clinical trials of transcranial stimulation. We touched on that a little bit last year, but using electric currents to get the neurons to fire. So there are groups looking at that specifically for patients with the progressive aphasias. And it's still very early days, but some of their results do look encouraging. So we will continue to bring you those updates. For potential disease modifying treatments, again, these are treatments that are aiming to actually slow that neurodegeneration, slow the cause of FTD or the toxicity to the cells, slow it down or stop it ideally. So across the world, there are a number of studies now. Most are for genetic forms of FTD, but not all. There is a new trial, very small. It's a phase 1 study, so very early testing of safety of a treatment that affects inflammation in the immune system for patients with semantic dementia. This is being led by the UC San Francisco group. But we spoke with them at the conference, and they do have funds to have patients travel to their center to participate as well. So if you know someone with semantic variant who might be interested in that, please let us know, and we can put you in touch with that team. There are multiple clinical trials now for the granulin mutation carriers, some running at our site that are multi-center that we're participating in, including antibody infusions, and then others at other sites in Canada, including one-time injections of viral vectors that carry a normal copy of the granulin gene to try and restore its production in the cells. And then we also heard about a new study coming of an oral compound for granulin, not in a clinical trial yet, but hopefully coming before long. For C9, there right now is the same antibody infusion trial that's running in granulin mutation carriers. That's probably just about to close enrollment. So we would hopefully know in about a year whether that looked to be helpful or not. And for the C9 mutation carriers, there's a study in patients with ALS of an antisense oligonucleotide. And again, I'll refer you to last year's lecture, but it's a really neat molecular therapy where you develop a mirror image to the gene mutation, and that mirror image molecule binds the RNA from the mutation and blocks it from being made into the mutant protein. So, you know, an approach that seems to have a lot of promise. They now have some very early data in the C9orf72 mutation carriers who developed ALS, that the therapy is reducing the levels of the mutant protein. So that was very exciting biomarker news. But we don't know yet whether it's actually slowing overall disease progression. So we'll be watching closely for those results. And then finally, for the MAPT mutation carriers, not studies that we know of just yet, but there are several in the pipeline. And because the MAPT mutation is a bit rare, the field is trying to organize a platform trial where patients could come in. There might be 3 different experimental medications. They might get put into any one of those 3. A smaller group would be randomized to placebo, but it would be a way to test those treatments with a smaller number of patients and try and figure out as quick as possible whether they may be promising or not. So I'll just leave you with this quote from the NIH Clinical Trials website. And if you want to look for clinical trials anywhere in the world, this website has a lot of them, most of them, particularly in the field of FTD, and has all kinds of great information. And more locally, if you're in Ontario or certainly Southwest Ontario region, we've got a big research team who can also help you navigate this, both studies going on locally or help to refer you to our colleagues elsewhere if it's a study that you're interested in that we're not running here. And Sarah Jesso is our longtime research coordinator who serves as our point person for research recruitment. So you'd be very welcome to contact her by phone or email anytime if you have any questions. And just thanks, lots and lots of names here because I presented lots of different aspects of our work, and it obviously takes a lot of people to accomplish it. So I thank everyone for your attention. Again, I realize we kind of jumped off from last year's talk, so this is maybe a little more technical, and welcome questions on what I've said tonight, or maybe things I haven't. And if we need to, I can pull up some slides even from last year to help explain things.
There is a question here, Dr. Finger. An attendee asks, can I publish some of this information on our site, in quotes, Canadians living with FTD? And then the participant mentions we have 235 families across 8 provinces.
Sure, yeah, no, absolutely. We would appreciate that. The young adult study I mentioned is being published in Brain very soon, so there shouldn't be any problem in showing that data, even though it's preliminary, and certainly we welcome the sharing of the information. And for anyone in Canada, you know, to contact us if they're not plugged into you know, local expertise as well.
I don't see any other questions. I'll keep an eye on it though.
All right, sounds good. Yeah, we'll wait and see if any other questions come through, but very interesting, Dr. Finger. I always love hearing the research updates, and it's so encouraging to hear about so many clinical trials that are you know, happening for FTD, because like you said before, there wasn't much going on, right? So it's, yeah, very encouraging. And very interesting. The apathy study I thought was very interesting. That one. Yeah. Catherine, any questions or comments?
No other questions at this point. Yep.
All right.
All right.
Before we let Dr. Finger go, I'll just take a moment to remind everyone that about our next webinar. So next Wednesday, November the 30th, in the afternoon at 1 o'clock, we have a lawyer from Siskins who's coming to talk about legal issues, so power of attorneys and capacity issues. So if you haven't already registered for that and you're interested, just you can send us an email or call in, and Natalie can register you for that one for next weekend or next week. All right, no other questions coming through right now.
One more. Hang on. Oh, just thanks very much. And one participant would like to see Sarah Jesso's contact information again. And this participant asks, what is semantic dementia?
Sure, yep. So semantic dementia is one of the 3 subtypes of FTD. It involves neurodegeneration, typically of the left temporal lobe. And the left temporal lobe is really important for language. I think of it like the dictionary of our brain. So it's where we store words and the meaning of words. And when that part degenerates, it's kind of like these words from our mental dictionary get erased. So patients have trouble with semantics or the meaning of words and objects. So, you know, maybe they're having a conversation and all of a sudden they say, well, you know, what is, I'm trying to think of an example here. What is a vacuum? So suddenly a word they knew before, they just aren't recognizing. And that gradually happens more and more often for more and more words. So it's called the semantic dementia or semantic variant primary progressive aphasia. And it comes under the frontotemporal dementias because it's degeneration of the temporal lobe. And it tends to be associated with that TDP43 molecular pattern, which is one of the FTD molecular disease patterns. As I mentioned briefly, it tends not to be a hereditary form. It tends to be only one person in a family, across many generations that ever would get that. So there's a lot of interest in, well, why do these individuals get semantic dementia? There's some subtle clues that maybe it relates to some immunity or inflammation, and is it related to other autoimmune diseases or inflammation processes in the brain, but it's not proven yet. But that's sort of the basis for that one clinical trial that's trying an anti-inflammatory medication to see if it might slow the course of semantic dementia.
One more question. The attendee asks, can we get a copy of criteria for diagnosis of FTD?
Yes, and so I don't think I have that slide in this slide deck. I'll see if I can quickly find it on this computer while we're talking, but absolutely. And I think that's coming from Danielle, who's probably pretty familiar with FTD. For those that aren't, it's progressive changes in behavior or language that affect disinhibition, impulsivity, empathy, repetitive behaviors or compulsions, sometimes some hyperorality, with relative sparing of memory, although we now know, at least 25% of patients might have some memory problems at the beginning as well. And it typically is associated with, shrinkage or decreased activation of the frontal and temporal lobes, and is, not accounted for by a stroke or other condition. And actually, I think I've got that criteria here. I'll see if we can share. And we can probably add that. Well, I guess if I share it now, it's on the slide deck, so that may work. So this is the criteria for the behavioral variant FTD, the most common subtype there. So you can see that the symptoms and that you need sort of repeated occurrences of these symptoms. The neuropsychological profile, so trouble with executive function tasks, so that's sort of sequencing, multi-step tasks, tasks requiring some inhibition. Most patients have loss of insight. And then, we grade it on severity or likelihood, depending on whether they have supportive imaging or genetic factors as well.
Just a few more questions, if you have time to go through them. This attendee asks, can you give me a synopsis of Lewy body? This person's parent was diagnosed with cortical basal degeneration in early 2021. However, since has been in long-term care, and staff comment on how the person's actions are reminiscent of Lewy body or possibly Parkinson's.
Yeah, and certainly we know clinically there can be a lot of symptom overlap between those conditions, so it's not too surprising that they're both maybe on the differential. Lewy body disease is, again, same molecule that builds up in Parkinson's disease. It's just more in the cortex rather than in the brainstem to start. And it tends to be in visual parts of cortex as well. So patients with Lewy body disease may have those visual hallucinations, usually well-formed, usually at the beginning not threatening, of people, animals, children. They often will have executive function trouble. So their memory might not be too bad, but they'll have trouble with more complicated multi-step tasks, trouble using the television remote, trouble doing their taxes, those sorts of things. As time goes on, they may develop some more Parkinsonism. So motor symptoms like Parkinson's disease, so a bit of a pill rolling tremor, slowness of their movements, kind of stooped posture, a shuffling walk, masked faces, so less expressive facial expressions. And often in Lewy body disease, the autonomic nervous system is affected. So they might have very variable blood pressure. Their blood pressure may be high, lying flat, and drops quite a bit when they sit or stand. In cortical basal syndrome, you can also get those posterior sort of visual processing parts of the brain affected. So that's where you can see some overlap. And in cortical basal syndrome, you can also get the same networks that would produce Parkinsonism. So you might have tremor, rigidity, slowness of movement, or trouble using an arm. So certainly those can overlap. And we know for sure that if somebody meets criteria for cortical basal syndrome, sometimes when we look at their brain, it's going to be caused by the Lewy body molecular pathology. Other times, cortical basal syndrome is caused by Alzheimer's type pathology or frontotemporal pathology. So it's really driven by just where those changes happen to be in the brain and less driven by what actual molecular change is there.
Okay. Thank you. 3 more questions. Is there a typical timeline for the progression of FTD?
There's a typical, and then there's quite a bit of variability. So, on average, if we look at the group studies for the behavioral variant, the time from really clear symptom onset, which is usually around the time of diagnosis, which is several years into symptom onset, usually from there to kind of advanced end-stage disease or death is around 7 to 9 years. But again, that's the average. So some patients progress more quickly, some more slowly. The semantic variant is a little different than the other subtypes. It tends to progress more slowly on average. So there, it might not be until 11, 12 years, even longer, that someone sort of reached the very advanced end stages. But we know, even some patients with semantic variant can progress more rapidly.
Thank you. Question, are there any people with dementia ever tested for heavy metals that could have affected them earlier in life? So I guess a known exposure to heavy metal.
Yeah, there's been a fair bit of research on this over the years. And for the dementias we're talking about now, there really have not been convincing associations with heavy metal exposure or, detection even, with the exception that manganese, which is a rare heavy metal, has been associated with some kind of atypical forms of Parkinson's disease. So that would be quite rare, in specific kinds of welding work and specific exposures. Exposure to heavy metals certainly can cause other neurologic problems. Lead, cadmium can cause neuropathies and other sorts of problems, but studies don't point toward heavy metals and Alzheimer's, Lewy body, or FTD, at least not at this point.
Thank you. Question. So if you have the C9orf72 gene mutation, can this be passed on as FTD or ALS?
Yes, and sorry, I didn't mention that. We kind of went to the advanced topics today. So yes, worldwide, the C9orf72 mutation is the most common genetic cause of ALS on its own, of FTD on its own, or in patients where those actually occur at the same time. There's, a lot of interest in, well, if someone has a C9 gene mutation, why would some people develop ALS and some develop FTD? And it looks like there may be some other genes that kind of influence which you're more likely to develop, but it's not established well enough yet to be part of the clinical, testing algorithms. It's hints that it might influence one way or the other, but we still need to figure that out. The geneticists need to figure that out further.
And one last question. Are all these conditions distinctive, as people seem to have characteristics overlapping many of the different labeled dementias?
Yeah, great point, and I would agree for sure. So at the clinical level, which is the symptom level, as Denise picked up, there's lots of overlap. You could have memory problems in Alzheimer's disease, but you could have memory problems in FTD or Lewy body disease or vascular dementia. Similarly, you could have behavioral changes in any of them. You could have hallucinations in any of them. So when we group them and try to make these distinct groups and partitions, it's somewhat based on the most common symptom clusters, but it's really also driven, as you saw early on, by what's the underlying molecular change. And the reason that the pathology and those molecular changes have really defined how we call the diseases is because there's this idea that these different molecular pathologies are going to need different treatments. That, alpha-synuclein building up into Lewy bodies is going to need a different treatment than amyloid and tau clumping up in Alzheimer's. So that's part of the reason that they are kind of in distinct groups. But that said, even at the molecular level, they may not be as different as we think. And we recognize now often Lewy body changes and Alzheimer's changes are both seen in the brain, and they might be influencing each other. And the same for stroke and Alzheimer's disease. So the boundaries help us to try and be talking about the same patients and doing research on groups that we can compare from one site to another. But yeah, we make it a lot more, black and white or distinct than it really is. There's lots of things that come in between.
Great.
That's all the questions.
All right. Thank you so much, Dr. Finger. It's always a pleasure having you. I look forward to it every year, and we'll anxiously await next year and see where the research is a year from now.
Oh, great. No, thanks so much for everyone's time and the invitation. And I'd say, please, send feedback to Tara and Catherine for next year so that we'll know sort of what would be most of interest and helpful to update on.
Yeah, absolutely. And we will email out a survey to everyone this week who attended tonight. So yeah, please give us your feedback if we have more specifics or things you want to learn more about next time.
Fantastic.
All right. Thank you and have a good evening, everyone.
Thanks. Bye-bye.
Just a comment. Thank you. And very informative and interesting.
Okay.
All right. Have a good night.